Hi, good evening. I'm Lonnie Stonich, the Executive Director of the Family Action Network, otherwise known as FAN, and I'm so excited to welcome you to tonight's conversation between Professor Eben Kirksey and Dr. Britt Wray. Thank you for attending. Today we celebrate Veterans Day in the United States, and we honor our country's veterans for their love of country and willingness to serve and sacrifice for the common good. So best wishes to all those. FAN is a nonprofit organization that presents a high-quality speaker series offered free to the public on a wide variety of topics, including human development, mental health, education, and social justice. We have over 100 videos of past events archived on our website and YouTube channel, so please be sure to explore. And now for my introduction of our two featured guests. Professor Eben Kirksey is a cultural anthropologist and member of the Institute for Advanced Study in Princeton, New Jersey. He holds a long-term position at Deakin University in Melbourne, Australia. In academic settings, he is perhaps best known for his work on multi-species ethnography, a field that uses innovative approaches to study human interactions with animals, microbes, fungi, and plants. Professor Kirksey has been published in Wired, The Atlantic, The Guardian, and The Sunday Times, and is sought out as an expert on science and society by the Associated Press, The Wall Street Journal, The New York Times, Democracy Now!, Time, and the BBC, among other media outlets. He speaks widely at the world's leading academic institutions, including Oxford, Yale, Columbia, UCLA, and the International Summit of Human Genome Editing, plus music festivals, art exhibits, and community events. Dr. Britt Wray is a science writer, broadcaster, and researcher focusing on synthetic biology and the psychological impacts of the climate crisis. She's the author of Rise of the Necrofauna, The Science, Ethics, and Risks of De-Extinction, and her forthcoming book, Generation Dread, and I hope she comes back for us, explores wide-ranging psychological and emotional responses to climate disruption, as well as what can be done to cope. She also produces a newsletter, which I've subscribed to, called Gen Dread, which dispatches weekly analysis about eco-anxiety, eco-grief, and other mental health impacts of the planetary health crisis. She has hosted several science podcasts, radio programs, and television shows with the BBC and CBC. She holds a Ph.D. in science communication from the University of Copenhagen and is a TED resident. It's my deep pleasure to welcome Professor Kirksey and Dr. Wray.
Thank you, Lonnie. Thanks for having us.
Thank you so much, Lonnie. It's great to be here with you, everyone. I'd love to kick us off tonight by explaining, firstly, how impressed I was when I read The Mutant Project, because it feels not as though it was written by an anthropologist, which Dr. Kirksey is, but an ethnographer and a historian and an investigative journalist really wrapped all into one. Eben has a really fantastic knack for being able to turn scholarship into captivating narratives that also capture close-up encounters at the vanguard of human genetic editing, which are fascinating and troubling at times to read. He also has a very, I would say, finely tuned radar for ethically complex social justice issues that gene editing technology creates and produces in the world. So I'm really excited to be here with you today, Eben, to dive into your work, as well as be here with all of you who are watching. So let's start.
And I should just jump in and say it's a real honor to have you with me here at this moment, and I'm really honored.
Oh, thanks. Likewise. So let's start with the book's dedication, because in the opening pages here, you dedicate the Mutant Project to Lulu and Nana. So who are they?
So yeah, Lulu and Nana are Chinese babies, twins that are now approaching 2 years of age, and they're basically born amidst much controversy a couple years ago. And really, they're not that much different from you and me. Just a single gene has been targeted in their genome for deletion, in theory, making them resistant to HIV.
And you say resistant to HIV. Are they in fact resistant to HIV?
Well, part of the complexities of the story that I tell are, kind of the scene where it all took place. So I kind of stumbled into the scene. I was invited to speak, as Lonnie said in the introduction, on an ethics panel in Hong Kong, where people were coming together to talk about, in theory, how these tools might be used. And this tool called CRISPR has been around for a while. And there was this idea that we all needed to get together and talk about it and debate, the issues before they became issues. So I arrived in Hong Kong and was totally jet lagged, and the news broke. And Dr. He was there on the couch trying to explain himself to other scientists. I learned about it in the elevator from another conference delegate. And basically, throughout the course of researching this book, I found out that the study was never done to determine if they were HIV resistant or not.
So it hasn't been verified. In order to go deeper into talking about this experiment that Dr. He conducted, let's just back up for a moment and speak about gene editing at large. What do we need to know about gene editing? Give us the equipment in order to understand your book, as well as detail for us, please, what CRISPR technology is.
So Britt, you and I first met at the first Gene Editing Summit, which got a lot less press. It took place in Washington in 2015. And at that point, CRISPR was more or less in the domain of speculation. People were talking about things that could be done with it. And it works similar to other tools. So there's other tools, zinc fingers, talons. What CRISPR does, what zinc fingers does, is it grabs a hold of DNA. So DNA is wrapped around itself in a double helix, and CRISPR basically grabs a hold of the DNA, produces a break. And in some ways, I think technical language describes it best. It produces targeted mutagenesis. In other words, CRISPR makes mutations. When the DNA is broken like that, basically the cell tries to repair the damage and usually doesn't get it quite right. So you might end up with an extra letter of DNA there. You might end up missing some. So basically that's what it does, is it produces targeted damage to the genome.
Okay. Targeted mutagenesis, and now we see the link to the name of your book itself.
Right.
Yeah. So one more stage setting question for you around CRISPR and human gene editing before we go into particular things you discovered while writing your book. Why would anyone want to do human gene editing in the first place? What are they setting out to achieve with this?
Well, I mean, there's health applications, right? So HIV is the one that we've talked about so far. There's a lot of gene therapies that are coming to market that are radically new treatments for cancer that could make, things like chemotherapy obsolete. But there's also whimsical applications that people are starting to explore. For example, it would be relatively easy, not necessarily with CRISPR, but with other tools, to insert new genes into people that would maybe make them glow green. So you could do that. That's been done in animals for a long time now. But I think we got to keep in mind that biology is very complicated and whole organisms are very complicated. So the average adult has 34 trillion cells, and each one of those cells has its own nucleus, chromosomes, and then the DNA packed into those chromosomes. So if you want to try to change an adult, you got to roll the dice as many times. Like basically, CRISPR does this targeted damage, but there's no way of really hitting all 34 trillion cells in a human body. If you start at the single cell stage or right after the first cell division, like with Lulu and Nana, you can produce, a genetic change that is going to be the same for that person as they grow up. But if you do it in an adult, the pattern is going to be, really unpredictable. Each cell is going to have its own idiosyncratic mutation.
So there are different types of gene editing then, ones in which we can edit just individual cells in an individual person, and other forms of editing in which you can edit the cells that go into creating a person early enough in their development that you end up then affecting changes in the entire human body, whether we're talking about editing sperm or eggs or embryos themselves.
Yeah, and if you do it at that early stage, then that change also gets propagated to future generations. So you're not just changing that individual person, but if they grow up and have kids of their own, those mutations will go on to the next generation.
Right. So that already raises a very interesting question. Do you think about the ethical valences around these two approaches to editing as being categorically different? Because one does involve passing genetic mutations through lineages and the other just involves affecting one individual person?
Well, for starters, I think it's important to recognize, like this word in the title of my book, The Mutant Project, is kind of charged. But, we've got to recognize that we're all mutants. We all have, from, anywhere from 40 to 60 original mutations that our parents didn't have. This is part of what makes us distinct, biologically unique individuals. So I guess in my mind, just thinking about the genetics, there's a lot of unknown risks about CRISPR. But if we're just thinking about the genetics, producing, if we could get a tool, and maybe it's not there yet, that'll do just a single mutation, I'm not too concerned about that going on to future generations. So I guess for me, some of the more pressing ethical issues relate to, who gets access to these technologies? Are they affordable? Who gets to use them? Whose dreams about the future are coming into contact with reality, and whose are just pure fiction?
Which is definitely where you put the focus of energy in your book in order to spell those out. And so we'll get into those. But first, I just want to take this broad kind of emotional stance at the field, because human gene editing is not an issue that people respond with shrugged shoulders or just a meh about. People respond with hope and optimism and adoration in the human capacity to be able to do something like cure genetic disease, for example, or they revile and disgust and anger that scientists would ever be so hubristic to think that they should be able to tinker with the human genetic blueprint in the first place. Of course, there are a variety of other emotions that could be invoked, like rage at the social justice issues that can also be provoked here. So I'm curious, before you started to do your deep research and writing for this book, what emotions, what feelings were driving you towards this field? And what were those feelings pushing you to try to find out?
Well, for starters, when I shop at a grocery store, I usually select the non-genetically modified food, knowing enough about how agriculture works in this country, and, the first line of genetically modified organisms, many of them created more ecological problems than they solved. I think one of my other big concerns going into the book was about eugenics. This idea of eugenics, literally when you break it down, means good genes. So, we've all learned about the Nazi death camps in history classes. Less well known are the sterilization campaigns that targeted gay people, that targeted the so-called feeble-minded. Even immigrants were targeted in states like California and Oregon. And I think one of the real dangers with these technologies is not, a narrowly defined set of medical risks. There are risks, and we can talk about those. But I think some of the real risks come as this technology interfaces with society. So, do we want to try to perfect the human species? Like, would this tool edit out certain kinds of biological diversity? Can we live with new kinds of diversity as these experiments happen, sometimes go awry? Are the children like Lulu and Nana who result from these experiments, are they going to be recognized as being fully human?
Those are all very valid points and really probing questions to arrange your whole book adventure around. And now that you're on the other side of this process, how do you feel about those initial questions? Should we be as concerned as you initially got the sense that maybe we ought to be, given that you have a much more intimate understanding of the knowledge, and I would have met a lot of players in the field who are pushing these projects forth? What really troubles you when it comes to these issues?
Well, I want to tell you about one of the first people I met who participated in a gene editing experiment. He might actually be with us listening tonight. So Matt Sharp is a HIV positive gay activist who's been campaigning for innovative and risky experimental medicine since living through the AIDS epidemic and watching many of his friends die. And Matt, he basically, first of all, it got me to step back from all the sort of heightened concern and excitement about these technologies. And I asked him, when the genetically modified cells, they took his cells away, they modified them, they put them back, they're trying to cure HIV. Were you scared? What were you thinking in that moment when, this infusion took place? And he was like, well, I was, kind of hopeful. It took about half an hour. I went back to work. And he told me instead, let me tell you about this thymus transplant that I had in the 90s. You know, they cut my chest open. It was really bloody. They took this fetal tissue in, they stuck it in, sewed me back up. It only stayed there for a little while. Then they took it back out. It didn't work. I felt like I was being, you know, ripped open. So, you know, I think for this particular community, communities of HIV positive gay activists who really have been pushing the boundaries, I think we can really regard people like Matt and someone else who I talk about in the book, Jay Johnson, as moral pioneers, you know, trying out these new technologies that the rest of us are really reluctant to try. So, you know, as I came to know Matt and Jay, I came to really see the risks as less about, you know, biomedical injury, but, you know, big business kind of taking these technologies, exploiting the goodwill of people like Jay and Matt, who are willing to sign up for these trials, but then ultimately, you know, producing powered, you know, profit-driven medicines, basically making a medicine that most of us can't afford. The price tags of these gene therapies are upwards of $400,000, and you know, a lot of us aren't able to afford those kind of treatments.
Is that issue of financial access what you mean by the exploitation that gets engendered by corporations getting involved in this space? Can you tell us a bit more about what you mean by that?
So part of Matt's story is, you know, putting his body on the line for this first in man experiment. And then this company that conducted this experiment, a biotech corporation in the Bay Area of San Francisco called Sangamo, they basically made a lot of money off of some promising results. Matt found that his immune system was repaired for the first time that he had an AIDS diagnosis. So, you know, Matt's cell levels reached a healthy level for the first time after he participated in this gene editing experiment. But those promising results produced big returns for the company on the stock market. But they basically decided that HIV was no longer the best lucrative opportunity. They decided to take their gene editing technology and pursue other diseases. So I guess, you know, in part in the book, what I'm trying to do is reframe these questions of risk, you know, to not treat risk as some narrowly defined biomedical problem, but to try to think about, you know, broader risks in society. You know, how might these technologies go awry in ways that aren't, you know, within the conventional paradigm of bioethics, where if you get somebody to consent to an experiment, then that is sort of certified as being ethical.
Right. And, you know, I've heard various researchers pushing at the vanguard of this field saying that, you know, there's really nothing unique about gene editing as compared to a variety of other shape-shifting biotechnologies that have appeared and then found their way into the reproductive clinic, for example, IVF, that has stirred a lot of public anxiety around what this means to change the nature of human relationship to nature and each other. Whenever there's this kind of new sophisticated tool on the table, we have the same reactions, we have the same questions from various forms of activism and social scholars pushing back on it. And this feels like deja vu. And really, when you boil it down, a lot of these worries are speculative and not tied to actual experiments or projects that are ongoing. And I'm wondering what your reaction is to that. Like, let's talk about some of the disability issues, for example, and CRISPR itself in ways that you think this might be uniquely threatening debates around ability.
Well, for starters, maybe before we get to disability questions, I'd like to back up and talk about, you know, the power of DNA in popular culture. So, okay, you know, since the 1950s, we've been told that this is the code of life, that this is kind of, you know, the molecule that makes us who we are. This is the secret of human nature. And, you know, as we are kind of getting further into the genetics and genomics of our species, you know, some of these older questions about nature versus nurture are getting resolved. So, you know, in the 90s, there's all kinds of speculation about, say, a gay gene, or, you know, about genetics and intelligence, or, you know, we're learning that some things are relatively easy to control with genes. You know, something like blue eyes is a single base pair of DNA that you can toggle back and forth, you know, blue eyes or not blue eyes. Something like height is incredibly complicated. Not to mention, you know, something as ephemeral as intelligence. There is no gay gene; it hasn't been found. But I think the homogenizing tendency of these technologies is very real. You know, one of the chapters of the book focuses on my friend Gregor Wolbrain. He's a biochemist and a theorist of disability. And he doesn't have a congenital condition. He doesn't have any disability as a result of genetics, but he identifies with people who do, and is worried that people like him are going to be edited out of existence. Basically, when Gregor's mother was pregnant, she took a drug called thalidomide for morning sickness, and as a result, his body's different than ours. And there's a really great film out there that I highly recommend. It's called Fixed, and in Fixed you can see Gregor very adeptly navigating the landscape and working in his lab doing biochemical research. But the movie really does explore some of these core questions about what are we doing as a species? Should we live with biological diversity, or should we try to enhance and optimize ourselves? And Gregor's a voice in that film that, first of all, comes out and says, I'm happy with who I am. Like people like me get treated poorly in society, but I like my body. I like to crawl wherever I can. I don't want to be normal. I don't want to walk. I might want to fly. He playfully will suggest things like that. And I think part of what Gregor wants us to think about is, can we live with accidents? Can we live with the mistakes of technology? And he's kind of living testament that it's not that there's anything unique and new about these new experiments. The experiments of the recent past have also had profound consequences on our diversity as a species. And I really like the way that Gregor thinks in not trying to reduce problems to any kind of simple analysis. He's not kind of pointing at the company that made the drug, thalidomide. He's not pointing at the government regulators for making a mistake, but he's pointing at society and asking us to think about the disabling treatment that people like him experience every day. So it's not a problem of the past, it's a problem of the present. So with CRISPR, will people like Gregor be allowed to exist? In a near future, if this becomes a safe, stable technology that's widely used in the reproductive clinic, will doctors let patients choose to have differently abled babies? And I think it's kind of up to us to decide. It's still, the future is still up for grabs.
I mean, that really begs the question then, as these genetic technologies advance, can we trust ourselves to simultaneously develop the social technologies to be embracing of diversity and difference? Is there a way to use the advancement of a field that's so powerful, such as gene editing, in a fashion that ensures an opening towards diversity rather than this homogenizing force you keep coming back to? Do you see any signs for how that would be possible to embrace that tension, that ambivalence?
I think for sure it can be used in that way. And that's really what this book is about. And I used the phrase moral pioneers earlier, and that comes from a book by Raina Rapp. And Raina was writing about amniocentesis, a very early genetic detection technology. So it used to be, if you were considered to be too old to make babies without biomedical monitoring, if you were deemed to be of advanced maternal age, your doctor would really put pressure on you to undergo amniocentesis, even though amniocentesis had the risk of miscarriage. Right. So in those early days, people were getting these diagnoses that their child would have Down syndrome. That was one of the early easy things to identify. And some of these women were choosing to have those children. Some of these women were making the choice, I'm going to kind of refuse what the medical establishment is telling me to do and have a differently abled child. And now what we're seeing, in doing research for this book, everyone's talking about CRISPR, but I learned about all kinds of other genetic technologies that are already in the clinic that are having those same eugenic tendencies. So again, eugenics, good genes. So now it's not just older mothers that are being encouraged to undergo genetic testing. Everyone is being, encouraged to do this, at least in the US and Europe and China. So now it's no longer amniocentesis, but a simple blood test. And with these blood tests, these prenatal screening tests, you can learn about a diversity of genetic conditions. And at least in this country, there's very little laws governing this. You can identify any potential genetic condition and have that be the basis for either terminating the pregnancy or starting some kind of therapy. So in a sense, I don't think that these questions are particularly new as they pertain to CRISPR. And CRISPR might help us really think about problems that are already out there that have slowly, steadily creeped into the clinic without many people noticing.
Are there any rogue players in the field who you think we need to keep an eye on in terms of how they are adapting CRISPR for these kinds of homogenizing forces?
Well, I mean, that idea of the rogue is, it's one that I also try to unpack in the book. Dr. He is painted by many as, China's Frankenstein, this monstrous scientist who, took risks that other people were unwilling to do. So part of what I've tried to do in this book is really kind of show who he is, but also I explore, some serious shortcomings of the experiment, which we can talk about in a little bit. But one of the people I also introduce readers to is a man named Zhang Zhang, who was one of the supporters of Dr. He. After these two twins were born, they traveled to a special medical tourism zone called Hainan in China. So in Hainan, it's kind of like Hawaii, but sort of built for medical tourists. They talked with the provincial government. They'd given a proposal to the governor's office, and the vice governor entertained them and basically was very supportive of this new initiative, offering land and money for a clinic. And on this proposal, they said that they wanted to do gene editing in the fertility clinic, as well as 3-parent babies, something that Zhang Zhang has already grabbed international headlines for doing, basically taking the mitochondria, sort of the powerhouse of the cell, and inserting it into the egg of the mother. He did that in Mexico and got slapped on the wrist by the US government. So Zhang Zhang, if you Google New Hope Fertility Clinic, designer baby, you can learn about what he's offering. And he's worked in Mexico. He's got experiments going on in the Ukraine. He traveled with Dr. He to Thailand as well. Thailand is a place that Dr. He was also doing CRISPR experiments and implantations. So, there are people who are still working on this, and, the technology is still there. And, it's not something that went away when this one particular individual went to prison. And I think we need to be leaning into this rather than pretending that it's not going to happen again.
Okay, so considering that there are developments, and of course, developments that we don't even know about could, of course, always be in our midst. We also need to consider the safety of these technologies, the efficacy of what they're meant to do, and track how that is being sharpened as more research comes out of the labs that are working with CRISPR and other human gene editors. But you do spend some time talking about, for example, why you don't like to call this gene editing. You like to call this genetic surgery where slips and cuts can cause real harm. So just could you tell us a bit about what the state of safety is with CRISPR tools right now, and how you think we need to consider that in the fold of risks that you're raising?
So for starters, metaphors are so important to understand what is going on here. I kind of explained how the double-stranded break works with CRISPR, but to understand how this technology works, I think we have to think very clearly about how we apprehend these messy molecular dynamics. So with gene surgery, you slip and you create an unintended injury. The metaphor of editing implies you type a sentence, you don't like what it says, or you made a typo, you go back and fix it. But CRISPR doesn't work like that. It can produce damage that's very difficult to fix. You can't just go back in with your editing software and clean up a mess. I like to use the metaphor of a drone, like a Reaper drone. So with a drone, you can give it coordinates, a GPS target, and it often goes to that target and hits the target. But sometimes it takes out the wedding party, and sometimes it gets the wrong coordinates or goes astray and makes a strike somewhere else. And CRISPR can work like that. It can produce targeted damage at a particular site, but you don't always know what you're going to get. A paper just published a few weeks ago showing that not only might you suddenly lose like 100 or 1,000 base pairs of DNA, you could maybe even lose a whole chromosome if you're using CRISPR. So the technology is really not there yet in terms of being safe to use. But that's not to say that it could be there. There's constant development in this field, and people are trying to develop new, more reliable tools. And I'm sure that they're going to eventually get something that works. If you go back again to the 1950s, I'm not the first person to say this, right? So Hannah Arendt in 1958 saw that scientists were already talking about recombining genetic elements from people of superior ability. You know, Hannah Arendt survived the Nazi Holocaust, and you know, she saw how eugenics produced very serious problems in her lifetime. And you know, these things haven't gone away; they're still here with us.
Presumably, many of the risks that society rightfully worries about can be mitigated by regulation, though no. What is the landscape of legal frameworks right now that support and hinder human gene editing, and how big are the loopholes?
So some places like India and Australia have total prohibitions. You can't do this kind of research there. The UK, interestingly, has a very good set of laws in place and active monitoring and regulation of this space. What we've seen in the US in the last 4 years under the Trump administration is a radical deregulation, a rolling back. So we used to have this committee at the NIH that regularly met. It was called the Recombinant Advisory Committee, or the RAC. And you know, ever since the 80s, they've been trying to do this precautionary governance work, anticipating, you know, you would have each university or institution would have their own local review committee, the IRB. And, you know, proposals that were deemed too risky or too novel were taken to the RAC, where a group of national experts would meet and debate and, you know, consider issues of social and economic justice at the same time they were considering the medical risks. But I was there when they ushered the first CRISPR experiment into the clinic, when the RAC gave the go-ahead. And you know, shortly thereafter, it was shuttered. There's been a new RAC opened up, but it hasn't met that much. It's sort of a modified committee. So we're really going backwards in the US. And you know, when I talked to one of the scientists in the UK, Helen O'Neill, someone who I feature in the book, she used the phrase, the horse is already bolted, when talking about the US. You know, there's all kinds of genetic technologies that are already being used in the clinic. And, you know, without sort of stepping into this gap, you know, there's so much going on in the news right now. Like, who has time to, like, you know, think about this and draft up a new law and actually implement it? You know, with all the different things in the news cycle, it's really hard to pay attention to something like this. But I think it's more urgent than ever. And if we don't, you know, sort of collectively deliberate about this, the laws are going to get passed behind closed doors. So another scene. So Britt, when we first met at that summit, somebody gave a presentation. There was a Q&A session, and a lobbyist got up and asked a question, and I followed him into the hallway. And you know, we were having this deliberative democratic discussion in this public forum. It was great. You know, all kinds of diverse people were there. You know, gay activists were there, patient advocates were there, transgender people were there, you know, alongside like the Pentagon and all sorts of unusual bedfellows. But as we were having this democratic discussion, there was someone out in the hallway on his laptop, quietly like writing to Congress and shaping the law in real time. So most of us don't have time to do that. And I think it's time, you know, we all need to kind of lean in to this issue, and because I think it really has broad implications for society.
Yes, and that points out a huge infrastructural issue we have. I mean, where is the apparatus that we can use to come together and collectively inform those policies? For example, sure, we have the human gene editing summits that have taken place, and a variety of groups have formed around the world that are trying to activate discussion in various localities. But we all have a stake in what happens with this technology. We all share part of the human genome. And yet it often feels like these decisions are being made without us, let alone without the, you know, perhaps most implicated stakeholders, like certain patient communities could be consulted in a different way than the rest of us and the general citizenry, and that would be appropriate. But could you just reflect on how we get past this challenge of constantly saying, yes, we need societal consensus. We shall not, you know, go past a certain line until we have that deep societal consensus, but there doesn't feel like there's much work being done to be able to ensure that we can ever get to that point of rich, sophisticated engagement on these issues with one another, upholding democratic principles. I mean, what do we do with that quagmire?
So I go back to these two people who I've already mentioned, Jay Johnson and Matt Sharp. And, you know, I think the rest of us have a lot to learn from this intergenerational political struggle that gay HIV activists have waged for the last 30 years over equity, over issues of, what is the research agenda, which kinds of diseases are going to be worked on here. And, this struggle came out of an organization, a grassroots collective called ACT UP. It was very much like Black Lives Matter or Occupy Wall Street. There wasn't sort of a central leader. It was just this distributed group that was motivated by just this shared sense, like we're all dying and we need to do something. And initially, ACT UP was, kind of an antagonistic organization. They would go do these really fun carnivalistic protests. So some were fun, some involved like, rainbow smoke bombs, like they stormed the NIH one year and all the scientists were coming out in their lab coats. They had no idea what was happening. And, all these men in all kinds of imaginative costumes were getting arrested. But then they also did some really dark, moving protests. Some of the iconic ones were taking the ashes of dead lovers and dumping them on the White House lawn. So over the years, as they made political progress with that struggle, they started to collaborate with their former antagonists. So ACT UP alumni now sit on government panels. They advise pharmaceutical corporations about what lines of research are worth pursuing and which aren't. And meanwhile, other advocacy groups around diseases are much less politically savvy, I think, both in terms of like the scope of, the political imagination, but also the effectiveness of the tactics. So my mother had cancer, and, I'm deeply excited about the prospect of not having chemotherapy anymore, right? So one of the potential benefits of gene therapy is these new cancer cures that could make, like losing your hair, like chemotherapy feels horrible. And we could have these new medicines. But, really, I think learning from those hard struggles and part of what Jay Johnson's key struggle has been, Jay is an African-American healthcare advocate, worked as a nurse for many years, cared for many people in the 90s who were dying of AIDS, and now works with an organization called Action Wellness. Their key struggle is about working with folks who might not have insurance, who might not have documents, who are getting discriminated because of their race, because of their gender identity, and trying to get medicine for them. So, Jay is confident if we can get, say, a cure for HIV, he thinks that he can do the effective political work to make that treatment available. And I think the issue of, creating children who are genetically modified is a separate one. And that I think, again, kind of circles back to some of these questions that are very important for the disabled community. So Sarah Katz has written a fabulous piece for Discover magazine about why deaf people don't want to be cured of deafness with CRISPR. There's many people who enjoy vibrant cultures that could get eliminated. Like if insurance companies, say, are demanding, like there's ways that power is working in these medical networks that is so important to think about. So I think that's where some of the key activist struggle needs to take place. It's imagining these near futures where a medical imperative is going to kind of enter the clinic. So I think if we go back to that amniocentesis example, there was kind of a preordained conclusion that doctors would expect. You get this diagnosis of your baby, like the doctor thinks that you should terminate it. Like there was really difficult struggles that many of these moral pioneers that Raina Rapp went through. And I think in the near future, if a deaf parent wants to have a child that's like them, there might be incredible pressure from society, from the medical establishment to make a change with CRISPR. And I think those are the kinds of things that we need to anticipate now and ensure that people are going to have those kinds of freedoms to maintain humanity as a diverse species.
And these questions also get sharpened when we consider the convergence of technologies, CRISPR with big data analysis, AI, for example, and genomic screening, having your whole genome sequenced perhaps while you're still in the womb, which is also a development that some people are excited about.
Yeah.
Yeah,
There's
perhaps that's
people
another
who are
webinar.
So as an anecdote, a friend, got his genes sequenced on 23andMe, and there's like social networks. There's like people who are basically using 23andMe as like a dating site to like identify perspective, like, ooh, they have good genes. I want to date them.
Yeah. Well, okay, so you raised some hope about working outside the system and antagonizing from, spaces that can address the inefficiencies of the public engagement structures that were offered, and then over time working to actually collaborate with those who are making policy and medical decisions, with your example of the AIDS activists. And that's really, it is a huge glimmer of hope here. But you do look at the outsider realm in your book of biohackers who are similarly applying pressure around some of these issues of access to the technologies, from creating their own levers upon which they can push on biotechnology, not that which is offered to them by institutional business as usual medicine. So just break that down a little bit for us. Who are the biohackers and how serious are their experiments?
So I've mentioned a number of times the price tags that are being attached to these new gene therapies. So Novartis produced the very first licensed gene therapy with this $400,000 price tag. Then a couple years later, they introduced a new one for $2.1 million for a single dose. And I think the biohacker movement really, in some ways, is kind of like the Robin Hoods, taking the knowledge and taking these potentially lucrative technologies and figuring out ways to democratize them. But the biohacker community is also very unruly, and there's not the same kind of set of rules that apply in these kind of anarchic collectives as you might see in a corporation. So part of the book chronicles kind of the rise and fall of one particular individual, Aaron Treywick, who kind of brought the principles of entrepreneurial innovation economy to the biohacking community and basically got pushed out. So, one of the key tenets for a lot of biohackers is this idea of open source technology, this idea that, we're going to use these tools and experiment with them and then make them available to others at cost or for free. And Aaron kind of entered this utopian space and managed to cobble together money. I kind of go into the darker side of where the money came from in the book. But ultimately, Aaron was starting to replicate some of the problems of experimental medicine. He was talking about going to South America and proffering these untested gene therapies on people who couldn't afford HIV medicine, basically taking advantage of very vulnerable people. So, I think the biohacking community is an interesting space of play and innovation, and a space where you don't see a clear line being drawn between medical uses of these technologies and uses that might playfully enhance diversity. One of the more imaginative and interesting things that I saw that this biohacking group that Aaron was part of working with was a gene that would let you see millions of new colors. So tetrachromacy is a condition that some women already have, and they can just see this diversity of new colors, and it basically involves a different kind of receptor on your retina. So to me, that's interesting and imaginative, and it's not about enhancing a soldier for the battlefield, but those imaginaries are there too. But I find that the hacker space is an interesting, unruly one, but definitely there's some problems to think about there too.
So I also want to talk about how you wrote the book, because I mentioned that it feels like it's written by an investigative journalist. You did a lot of sleuthing. You went to China 3 times. You even went to the rural village where Dr. He grew up to capture juicy details that led to the famous experiment that produced Lulu and Nana. So Tell us a bit about that process and what it was like for you to really go that deep in order to get some insider details. And as I understand it, you actually have some details about the experiment that no other journalist who has been capturing information as it's developed around this experiment has been able to get.
Well, for starters, before I did that stuff in China, I went really deep in the microfiche in the New York Public Library. So the initial start I had with the deep dive into this research basically started with HIV positive activists, the ACT UP community. And, you know, when we met Britt, again, CRISPR was in this realm of kind of hope and speculation. And these experiments had been done with this earlier technology, zinc fingers. So I went into the archives trying to figure out the history of HIV positive activism and experimentation. And as I was in the archives, microfiche kind of makes me seasick. It like goes across the screen. I found some people on Facebook who were still alive from the ACT UP movement. And they, within moments, put me in touch with actual participants in the trial. And, you know, Matt Sharp happened to be in New York that weekend, and he very generously shared his story with me. And I think, you know, when I gave my presentation in Hong Kong, again, at this moment of extreme controversy, I talked about the earlier experiment. So the experiment that Matt Sharp participated in, that was targeting the same gene that Dr. He targeted in these two babies. And it was basically the same goal, you know, producing a cure for HIV. And I also talked about the problems that, you know, participants in the trial experienced. There wasn't long-term follow-up, at least in Matt's cohort. You know, there's not rigorous attention being given to the safety of these things for the long term. And so some of Dr. He's employees, or one key person in particular, Ryan Farrell, who I name in the book, was in the audience and heard me speak. And I think he saw that I brought a critical eye to this and that I wasn't sort of demonizing a particular individual for a monstrous experiment, but that I really wanted to understand how, you know, investment capital, how the Communist Party of China, how these really complicated dynamics of this city called Shenzhen played into the experiment. So, you know, in doing the research, I found that in many ways, what he did was not exceptional. So Shenzhen is a place where China opened to capitalism in the 1980s. And it's a city that really aspires to embrace the future itself. The motto there is similar to Silicon Valley. You know, Silicon Valley move fast and break things. You know, it's an entrepreneurial ethos where you sort of do the experiment, you get results, and then you sort of seek recognition from Beijing after you actually do the experiment. So, you know, that was kind of the cultural scene that he was operating in. The book, yeah, basically goes into some of the ways that I, you know, managed to slowly build this story. But, you know, after I started to get access to some of these documents from the lab, some of the original data, some video recordings that were made of the participant consent process, and I started digging really deep, I learned something that hasn't been reported yet. So Dr. He announced this experiment to the world on YouTube, and he claimed that 2 healthy babies emerged from this experiment, Lulu and Nana, came crying into the world as any other, is kind of my paraphrase of his announcement. But when he recorded those videos, the babies were actually in the hospital. They were in the neonatal intensive care unit. So part of this book really goes deep into, you know, what life was like for Lulu and Nana in these first moments of life, and also the complicated stories of their parents who were living with secrets of their own. Their fathers were HIV positive, and part of what motivated them to seek this CRISPR experiment was an inability to safely make babies. In China, if you're HIV positive, basically you can't avail yourself of normal reproductive technology that is widely available there. It's just national policy. You can't basically use IVF, which would protect your partner and your child from the HIV virus, and you can't use a technique called sperm washing. Yeah. So they signed up for the experiment because they were trying to do the right thing. These parents really wanted to have a baby that would never have to experience the social stigma associated with an HIV infection.
And so what's next for Dr. He? You mentioned, of course, prison, but what have the consequences been? What have the ripple effects, manifested as?
Well, I think for him as an individual, I think it's still too early to say. He's been in prison during the coronavirus epidemic. So as I report in the book, early on, nobody knew where he was. He was rendered to a black site, and at moments, he was being confined in a facility where he couldn't even sleep on the floor. The prisoners had to take turns sleeping. I think for the field, what we're seeing is at the same time, the government is funding new initiatives along these lines. Embryo editing continues in China, but there's a lot more oversight and regulation. So the day that Dr. He was fired from his university position and detained by security forces, the president announced that there would be new regulations for gene editing. And he wasn't putting a stop to it, just calling for the new ethics committee to be formed and for universities and other institutions to have a lot more oversight as these experiments move forward. So it's not going away. It's just being done with a lot more government oversight right now in China.
I know we're going to wrap up soon before we head over to the after hours, but I do want to ask one question that came in before the session from an audience member, and that is, given the current inadequate rights and inadequate protections of LGBTQ people, what should the legal and civil rights, including reproductive rights for genetically modified humans be?
Well, I think this question about the rights of the genetically modified child is incredibly important. When this experiment first happened, all sorts of horrible language was being mobilized, and in some ways it repeated the language that was used 40 years earlier. So in many ways, this is history repeating itself. So Louise Brown was doing a book tour the year that Lulu and Nana were born. Louise Brown was the first test tube baby, the first child who was produced during IVF. And when she was born, everyone from the Pope to the scientific community condemned the experiment, and similar language was used. She was called a Frankenbaby. She was called monstrous. And Louise Brown, her whole life has been engaged in the struggle to be seen as normal. And now that we have millions of IVF babies worldwide, we're slowly starting to recognize, like, okay, this person isn't that different from us. And I think, for these two children who you asked about with your very first question, Lulu and Nana, that's going to be something that they might have to face later in life. And I think one key difference, and I think this is a big ethical difference between Steptoe and Edwards, who were involved in the Louise Brown experiment, and Dr. He, it was a media spectacle at the hospital when Louise Brown was born. She was a public figure from day one, and the identity of these two children has been protected. Their parents have been protected. And we can talk more in the discussion about the dynamics within Dr. He's lab, but I think that's one thing that they got right. These children might not want to participate in future experiments. They might want to just be ordinary preschoolers and ordinary high schoolers and, have a normal job. This is what Louise Brown is doing today, right? She works in a shipping facility in England. So, they might not want to be exceptional. And I think, again, this is something that the lab did right in protecting their identity. And I think not only do we have to guard their rights, we have to protect them from unwanted scientific experimentation. So right now, many in the Chinese government and the international scientific community are clamoring to do new research on these two young girls. And I think that needs to be part of the ethical debate. We as a broader civil society need to weigh in on that critical issue.
Now, Lonnie has joined us. Hi,
go ahead.
Lonnie.
Did you have another question, Britt? Go ahead. If you had one more, go ahead.
I was just going to ask, whether we like it or not, human-created mutants are here to stay. They're with us. And we have a variety of things to be aware of, which your work helps us think through. But what would be your dream when you think boldly and take a step back and gaze at everything that you've collected about this field? What is the dream about the justice-oriented future that we can bring forth with this technology in a way that embraces it rather than just shuns it away?
I think opening up the field of experimentation for, more interesting experiments, right? So the concluding gambit of the book goes to my mentor, Donna Haraway, who famously in the 80s wrote the Cyborg Manifesto. So in those days, people were afraid of computers. They were afraid of this thing that people were starting to talk about. DARPA had created something, these connections between computers that later became the internet. And Donna, in that essay, also writes about genetic engineering, and she writes about feminist science fiction. And some of the characters in these feminist science fiction stories would give a standard bioethicist the heebie-jeebies. You've got Superluminal, a novel by McIntyre that talks about Orca, this genetically modified human who frolics underwater with whales. And Donna herself writes about ways that we might modify ourselves, not for human health, but to protect the well-being of other species. So she writes about what it might be like to modify yourself to better empathize with another kind of life. In her case, she's writing about monarch butterflies. So I think, some playful, risky engagements with these technologies are also warranted. Again, the medical risks are going to be worked out, and we are going to probably see these homogenizing tendencies. So I guess maybe an opening question for this after hours discussion is, what kinds of biological diversity do we want to see in the future?
Thank you.
Okay, so I'm going to remind folks, if you've heard about this after hours a little bit, so it's a great chat is two-way, and everybody's faces are on the screen, and it's a great conversation. We put some links in chat about buying a copy of Eben or Britt's book. Do support them, support independent bookstores. Let's get those sales during COVID. It can be really so hard. I'm going to ask a couple quick questions here. I see a couple questions from a couple individuals that I know will be in after hours, so I'll leave that to later, and I'll instead pivot to a couple others. So Sophie Lewis asks, hi, Eben and Britt, thanks for this event. As a fellow anti-racist cyborgian, repro-utopian, could I ask, Eben, that you comment on the relation of the term mutant to the term monster?
Interesting. So this is something I engage with in the final moments of the book, and this is a conversation that Sophie and I have been having, over the course of a couple of years. So there's a lot of writing. I mentioned Donna Haraway just now. There's also biological theorists like Stephen Jay Gould, who has this classic essay called Hopeful Monsters. And Gould is writing against the dominant trends in evolutionary theory to think about the surprising possibilities that sometimes emerge with new forms of life. So, Gould is was famously the colleague of E.O. Wilson at Harvard, and the two of them would apparently have fights at the water cooler. And, E.O. Wilson is about kind of fitness, and evolution kind of advances as beings kind of optimize themselves. And Gould was about the uncanny and quirky surprises that sometimes emerge and create new kinds of biological diversity. So, kind of thinking and writing with Gould in mind, with Haraway in mind, also with Jacques Derrida, who insists that we kind of open ourselves to the lively possibilities of monstrosity. That's kind of the spirit that I conclude the book with, is just, let's not try to perfect ourselves. Let's think about new imaginative futures.
And then I have, let's see, we're looking at time. We have time for one short more question. I have a question about the racial aspect of this, quote, race. Can you imagine one day the handful of rich, monopolizing the technology and creating a new race of their own? They live longer, healthier, taller, stronger, and perhaps whiter. That's from Jing Fan.
So yeah, race is a big part of my book. And one of the findings was that the hospital in Shenzhen, China, that gave ethics approval for this experiment by Dr. He has a plastic surgery clinic where routine skin lightening treatments and Asian eyelid removal surgeries are performed. And if you kind of look at the history of cosmetic surgery in East Asia, it's a story of colonialism. It goes back to the Korean War when American doctors offered this surgery of eyelid removal to their counterparts who helped out during the war. And it's really caught on. And there's a particularity to whiteness in East Asia. Blue eyes or blonde hair would mark you as a foreigner, as an outsider, someone who's not entitled to citizenship and national benefits. But I thought it was remarkable and very telling that the values driving this experiment, it was profit, right? And values of beauty that are connected to these ambivalent legacies. How might we think about new biologically diverse futures where there's a room for brown people and Black people? This is, one of the other chapters is in Indonesia, where a transgender artist named Tamara Pertamina asks that question. With CRISPR, will there be room for brown and Black babies in the future? This is something she's really concerned about, and something we can talk more about in the after hours conversation.
Well, and that's quite a sobering note to end on, an important note. I want to thank Britt so much for such careful presentation and preparation for this interview. I'm sure Eben appreciates that as well. Thank you both for joining. Thanks everyone for tuning in.